Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
Picture this. You're surrounded by everyday things. Maybe your nonstick frying pan or your raincoat. And inside lots of these items, substances that could basically stay in your body, well, potentially forever.
And with effects we're still really trying to figure out.
Exactly. We're diving into the world of per- and polyfluoroalkyl substances today, often called forever chemicals.
You've brought a couple of interesting pieces here, a health news article and a pretty detailed scientific paper. And the big issue, like that news piece points out, is just how widespread these things are. Almost unavoidable.
Right. But there's still so much we don't know about what they actually do to us long term. So our mission today is really to unpack this. Given how common they are, what's the latest on the health impacts? And crucially, are some people maybe more at risk than others?
And to get at that, we're focusing heavily on this really neat study out of Duke University.
Okay.
They used these tiny little worms, Canarhabditis elegans, to look into the toxicity of 13 different per- and polyfluoroalkyl substances.
Tiny worms. Okay, you might be thinking, how does that relate to people? But apparently, these worms share quite a bit of genetic and biological makeup with us. Surprising, right?
It is. It makes them valuable little models for this kind of research. So the Duke team had a couple of main goals. First, see how these different chemicals, some older ones, some newer replacements, affected the worm's growth.
Okay, growth is a measure of health, basically.
Sort of, yeah. A basic indicator of toxicity. And second, they really wanted to know if the worm's own genetics, their different strains, made a difference in how sensitive they were.
And that's key because people are genetically diverse too.
Precisely. It gets at that question. Could the same exposure affect people differently? Maybe your genetic makeup matters. That's a huge implication for how we think about risk. It's not necessarily one size fits all.
Understanding that potential vulnerability and just the risks in general feels pretty important then. Helps you make choices, maybe push for better safety information or rules.
Yeah, definitely. So let's dig into how they actually did the study. It was quite systematic.
Okay.
They took different genetic strains of these C. elegans worms and exposed them to various doses of 13 specific per- and polyfluoroalkyl substances.
13 different kinds.
Right, and they chose them carefully to get a mix. Different structures, some had long carbon chains, some short. They looked at different functional groups attached, things like carboxylic acid, sulfonic acid, sulfonamides, even ones with ether linkages.
So like different flavors of these chemicals almost.
Kind of, yeah. Different building blocks attached to that main chain. And it turns out those differences, like having a sulfonamide bit, could make them more harmful.
Interesting. So what did they measure after exposing the worms?
After 48 hours, so 2 days, they measured the worm's growth, simple as that, and compared it to control worms that weren't exposed at all.
Right, to see the difference.
Yeah, to see the impact. And they calculated a specific value. It's called the EC50, effective concentration 50%.
EC50. What does that mean in plain terms?
Think of it like this. How much of the chemical does it take to slow down the worm's growth by half, by 50%? A lower EC50 means the chemical is more potent, more toxic. You need less of it to see that big effect.
Got it. Lower number, more toxic.
Exactly. It's a standard toxicity measure. And like we mentioned, they didn't just use one type of worm, these 12 different strains, all with slightly different genetics.
To see if that genetic background mattered.
Precisely. Just like people react differently to things.
Yeah, makes sense.
And they did one more thing for 2 specific chemicals, GenX, which is a newer one, and perfluorooctanoic acid, an older one people might have heard of.
Okay.
For those 2, they actually measured how much of the chemical got inside the worms. That's called bioconcentration. How much does it build up?
So not just the effect, but how much actually accumulates.
Right, because even if something doesn't immediately seem super toxic, if it builds up in the body, that could mean trouble down the line, like how things can build up in us.
Okay, that's the setup. Seems thorough. So what did they actually find? What were the big takeaways?
Well, first off, for all 10 chemicals where they could actually calculate that EC50 value, they did stunt the worm's growth.
So they all had some negative effect at certain levels.
Yes. Confirms a biological impact. But what was really striking was the huge range in toxicity.
How big a range?
Get this. One chemical, perfluorooctane sulfonamide, was over 1,000 times more toxic than another one, perfluorobutanoic acid.
1,000 times.
Right.
Seriously.
Yeah, massive difference. It really tells you can't just lump all these perf and polyfluoroalkyl substances together.
Wow. Okay, so some are way, way worse than others, at least in these worms. That seems important for, like, prioritizing which ones to worry about most.
Absolutely. And they saw patterns linked to the chemical structure, too. Generally, the longer chain chemicals, they tended to be more toxic than the shorter chain ones.
Okay, that kind of fits with other things we've heard, right? Longer chains might stick around longer or interact differently.
It seems that way. Another key structural finding, those chemicals with the sulfonamide functional group, remember perfluorooctane sulfonamide, the really toxic one?
Yeah.
Well, chemicals with that sulfonamide bit were generally more toxic than ones with carboxylic or sulfonic acid groups, so that specific chemical feature seems important for toxicity.
That's really specific. So the exact type, the exact structure really, really matters.
It absolutely does. And now, the genetics part, this is really interesting. They found that the different genetic strains of worms, they showed different levels of sensitivity to the same chemical.
Uh-huh. So it wasn't just the chemical, it was also the worm's own genes.
Exactly. This is basically the first direct proof that an organism's genetic makeup can influence how susceptible it is to these specific chemical toxicities.
That feels like a really big deal.
It is. They even identified specific strains, like one called DL238 was often more resistant. Another, CB4856, was often more sensitive to certain ones.
So just like people, some worms were naturally more vulnerable or more resistant based on their genes.
That's the strong implication, yes. And one last finding on those two specific chemicals, GenX and perfluorobutanoic acid.
The ones they measured accumulation for.
Right. They found GenX, the replacement, actually built up inside the worms similarly to the older perfluorobutanoic acid. But interestingly, GenX was less toxic in terms of stunting growth in this study.
Huh. So it accumulated just as much, but caused less immediate harm.
In this specific measure, yeah. It suggests accumulation doesn't automatically equal toxicity level, or maybe the toxicity works differently. It's complex.
Okay, let's unpack the implications then. This genetic variation piece seems huge. What does that mean for us, for people?
Well, it's the first real evidence suggesting genetics could play a role in and polyfluoroalkyl substance sensitivity. If that holds true for humans, and the shared biology makes it plausible, it might start to explain why, some people exposed to the same environment get sicker than others.
Right. It wouldn't be a simple cause and effect for everyone.
Exactly. It suggests individual susceptibility could be a real factor. Maybe one day, understanding someone's genetic predisposition could be part of assessing their risk.
And what about the structure? You mentioned how much that mattered.
Yeah, the study really hammers home these structure-activity relationships. It's not enough to just label something a forever chemical. You need to know which one. The chain length, those functional groups, they drastically change how harmful it might be.
So that information is critical for regulators, right? To figure out which of these thousands of chemicals need the most urgent testing and attention.
Absolutely vital. We can't treat them all the same.
We really need to know the specifics.
And the study also validates using these C. elegans worms. They turned out to be a pretty fast, efficient way to screen lots of chemicals and look at the genetic angle.
A new approach methodology, they called it.
Right. It could potentially speed up identifying harmful perlacea and polyfluoroalkyl substances compared to, say, slower animal testing. It's a way to get initial toxicity data much faster.
Sounds promising for tackling this huge backlog of untested chemicals.
It does. And finally, that comparison between the older chemical and its replacement, GenX, it showed that GenX was less toxic for growth, even though it accumulated similarly. But the worm's genetic responses to GenX and perfluorooctanoic acid were different, suggesting maybe they cause harm through different biological pathways.
So just swapping one chemical for another, even if it looks a bit better on one test, might not be the simple fix. It could have different problems.
Exactly. The replacements aren't automatically safe, and they might act in ways we don't expect yet.
Okay, but we need to be careful here, right? These are worms, not people. What are the limitations we need to keep in mind?
Oh, absolutely crucial point. Worms are not humans. While they share biological pathways, they aren't identical. These results are super valuable pointers, but they need follow-up.
Follow-up in mammals and eventually human studies.
Yes. To confirm these effects actually translate, think of it like studying a bicycle to understand a motorcycle. Similar principles, maybe, but definitely not the whole picture. This is a starting point.
Right, not the final word.
Definitely not. Another thing. This study focused mainly on growth inhibition.
Just one effect.
Exactly. We know from other research that these chemicals might have a whole range of effects on the liver, the immune system, development. This study didn't look at those other potential health outcomes.
So slowing growth is just one piece of the potential harm puzzle.
Correct. And lastly, they looked at 13 chemicals and 12 worm strains. That sounds like a lot, but there are literally thousands of per and polyfluoroalkyl substances out there.
Thousands. Wow.
And way more genetic diversity even within C. elegans, let alone humans. So while it's a great start, we need more research on more chemicals and broader genetic backgrounds to get the full picture.
More worms, more chemicals, more research needed. Makes sense.
Yep. Now, what did the researchers themselves say? They really stressed the urgency here.
Urgency about what?
About understanding the toxicity of this vast number of chemicals currently in use. That news article mentioned, over 14,000 of these substances? Many have never been properly tested for health effects.
14,000 untested chemicals we might be exposed to. That's kind of terrifying.
It's a huge knowledge gap. The researchers also highlighted the potential of using this kind of genetic insight, the kind they saw in the worms.
How so?
Well, maybe down the line it could help identify individuals or even whole populations who might be genetically more vulnerable to specific per- and polyfluoroalkyl substances.
Leading to more personalized risk assessment or protection.
Potentially, yeah. It could be a real step forward for public health protection if we can figure that out.
That would be significant.
And finally, they emphasize needing to understand the mechanisms, how these different chemical structures actually cause harm. If we understand the how for different structures, we can get much better at predicting risks for untested chemicals and regulating them more effectively.
It's about getting down to the nuts and bolts of the toxicity.
Exactly. So it all leads to this kind of provocative thought, maybe, for everyone listening.
Yeah.
Given that most of us probably have these chemicals in our bodies, and knowing now that our individual genetics might play a role in how they affect us, what's our responsibility here? To understand potential vulnerabilities, to push for more thorough testing, better regulation?
That's definitely something to chew on. So if people want to dig deeper after hearing this, what should they do?
Well, they can certainly look up the study itself. The paper is titled Structure-Specific Variation in Per- and Polyfluoroalkyl Substances, Toxicity Among Genetically Diverse Caenorhabditis elegans Strains. It's in the journal Toxicological Sciences.
Okay, we can point people to that.
And also, maybe look into reports from environmental health groups. See what's known about contamination in your own area.
Yeah.
Local information can be really eye-opening.
Good advice. And the final takeaway message.
Stay informed, ask questions about products, about what's in them, and, support efforts to get a better handle on these forever chemicals. Knowledge really is power here for protecting ourselves and the environment.
Thanks for tuning in to the Health Headliner podcast by To Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.